everyone, and welcome to today's webinar, AUC, Analytical Ultracentrifugation Capabilities for the Analysis of Gene Therapy Products. I am Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit www.beckman.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I also want to mention that there is a survey at the end of the webinar, so if you could please take a moment and submit your responses when prompted to do so, that would be greatly appreciated. I'd like to now welcome our speaker, Dr. Alexander Bepperling, Senior Manager, Analytical Characterization with Sandoz. Dr. Bepperling, you may now begin your presentation.
All right, thank you very much for hosting me today. For the introduction, I will also sharing my video. But to devote the bandwidth totally to the audio, I will switch the camera on during my presentation. So welcome everyone. Just before I start with the actual content, I will just show you a short disclaimer just in case you are wondering. A month ago, Sandoz has become a standalone company, been approved by Novartis and also published before the spin-off. So to be on the safe side. The work was done by two members of my team. One of her, Giza, was working on the lipid nanoparticles, and Janine working on the AAVs I'm going to show you today. And I'm assuming before we start that you all know what an AUC is and also what a standard sedimentation velocity and sedimentation equilibrium experiment is. So I won't go much into that detail, but I will focus a little bit more on the AUC data analysis and then jump right into two case studies. So AUC, sedimentation velocity, especially as you may know it, you can fit your SV data on your desktop computer like in SetFit, or you can do it with a supercomputer using Ultrascan and get a size and shape distribution. And in the case of, or in the simplest case, you have a single species in there with a sedimentation coefficient and a single value for the frictional ratio. This is an excellent method for the quantitation of aggregates, and the resulting molecular weight is accurate for the main species. But what if multiple species is present? You may have encountered that. For example, I'm showing here you a novel myokine, which is intended to stimulate muscle growth. And when you purify that, and you know that from sequence, it's an intrinsically disordered protein. And when you elute it from the sec column and have sec moles measured, it's a 220 kilodalton oligomer and a broad peak. In AUC, if you just do the, let's say, CFS analysis, you also see, okay, there's a single species, or presumably not a single species, but a single peak in there. And the question is, can we resolve that peak? And the answer is yes. Some of you may employ that. It's available for, I would say, 15 years right now. The two-dimensional spectrum analysis, where you do not have only an S value with a single F over F0. SetFit also allows you to do that. In Ultrascan, it's automated and performed on a supercomputer. You extract all non-zero solutes. You perform a series of grid shifts. And in the very end, you can also perform a Monte Carlo analysis and end up with a two-dimensional spectrum of shape and size, or size and shape, you may name it as you like. This may take some time, as said before, and when we apply that to the example I've shown you before, to this myokine, you see there are, this is measured at 0.2 mg per mil because it's an intrinsically disordered protein, which may behave non-ideal at higher concentrations. You see an array of at least 4 species and some larger stuff and perhaps a little bit of a degradation product. And when you zoom in there and look at the S and F over 0, and the molecular weight, you can see 4 distinct species. And if you go for the molecular weight, the monomer molecular weight is 13 kilodalton, and just multiply it by the oligomeric states and look what's in there. You see here a remarkably consistent 4 species in there, which correspond to 9, 12, 15, and 18 subunits. This tells you already quite a lot. So 2DSA allows you to determine the molecular weight, and thus, in this case, the oligomeric species. You may already assume, okay, the basic building block of this may be a trimer, because the difference between 9, 12, 15, and 18 is 3. And as said before, the F over 0 of about 2.2 indicates you that you have a disordered protein. So this is what you can do if you have a single, let's say, component in there, so just protein, for example. And in the case of cell and gene therapeutic products, you usually have at least 2 components. In the case of AAV, you have a protein and a nucleic acid. In the case of lipid nanoparticles, you have a bunch of lipid species and also nucleic acid. And in order to test how it would look like if we perform a multi-wavelength AUC experiment on that, and if we can spectrally and also hydrodynamically separate that, we tested it on the Optima AUC with a model system. I just took a DNA ladder and spiked it with 50 microgram of an IgG1 and collected a multi-wavelength experiment. And this is just for the A260 trace. You can see here in green the ladder, it's nicely resolved, the species, and in red you see it's perfectly overlaying for these species here. But for this peak, you have an additional increase in intensity, which is your IgG. I performed the experiment here in green first, and then choose deliberately a DNA ladder where you have exactly at the position, or very close, as close as possible, of the antibody, which we knew has an S value of 6.5, also a species, so that you really cannot hydrodynamically say, okay, this is protein, this is your DNA. And then we performed a multi-wavelength experiment and collected a full range of 30 wavelengths, ranging from 230 nanometers to 290 nanometers in 2 nanometer increments. This is the whole data set here on the left-hand side, and only 3 selected wavelengths to illustrate what it looks like. If you look at individual wavelengths on the right-hand side, red is A230, blue is the A280, and in between you have in green the A260 signal as before. And when you analyze now the data set with all the 30 wavelengths in a global fit, you can actually see pretty nicely. This is the view just if you look from the side, like a continuous C-S distribution. And this, you see here, you have a bunch of species in there. They have, most of them have a maximum here at 260, which means it's nucleic acid. And then you have a second species in there. I will now rotate it a little bit, where you can see this species has a 260 maximum, and then very closely adjacent to it, you have a species which has a maximum at 280 around, and then it has a backbone absorbance going up here at 230, which means it's a protein. So you can indeed spectrally deconvolute 2 species, even if they are very, very close in terms of size and S value in this case. So with that under our belt, we went to AAV, Zolgensma, the product, with empty spike capsids, because the full product itself contains very little empty particles. And we have been analyzing AAVs before with single wavelengths, and now we were just looking for a comparison of the conventional single wavelength AUC with multi-wavelength AUC, and also with some other kinds of experiments I'm going to show you later on. Just to bring you on the same side, the empty capsids, or the reason why this is so important, and it led to a tremendous increase of AUC work over the last years is the empty capsids have zero therapeutic effect because there's no nucleic acid in there, but of course they may still stimulate immunogenicity, so they are an unwanted product or side product within your formulation. Okay. If you look at the standard sedimentation velocity experiment, which we have been conducting at back then Avexis and then Novartis cell and gene therapy, in Mengish in Slovenia, for example, you see here in this panel the sample I've described to you. It's a Zolgensma product spiked with a certain amount, roughly 20% based on the A230 trace with empty particle, with just empty particles from the purification process. And what you can see here, of course, your different wavelengths give you very different answers. In red, you see you see a huge signal here for A230 for the protein, empty capsids, while when you look at the same peak here at 260 and 280, it's much smaller. And the question, of course, is always, what's the correct answer? We can, of course, use just a single wavelength, but as you can assume, these will give you grossly different answers. What you can try is you can calculate for the protein, and you can also measure that with the empty particles, and we did that, and also for the nucleic acid, extinction coefficients at various wavelengths. You can also use a program in Ultrascan implemented where you globally fit from a concentration series a internal absorbance spectrum with molar extinction coefficients. When you do that, you can improve the consistency between your wavelength, because what you can do is then you can calculate the titer in percent, so the capsid ratio, you see that here on the right-hand side, becomes a little bit more consistent compared just to the raw peak area, but still you see here in the bars, there is quite some variability in there between empty here in blue and partially filled in orange and full in here in gray. So the question is, can we do it better? And as you may suspect, multi-wavelength AUC may be a solution because you have a matrix of extinction coefficients there, which may also account for hyper and hypochromicity, which may be the reason for the still slightly different peak ratios, even if you apply extinction coefficients. So we did the same as I've shown you before for the DNA ladder plus IgD example. We collected a set of 30 wavelengths on the same sample. And already from looking at the raw data, you may see, okay, here this small contaminant or degradation product on the left-hand side, this may be protein plus DNA. It's steeply decreasing from 230 to 290, so it may be a mixture of quite a lot of things. It's very close to the buffer peak also. Then you have something here at 17S, which may be likely the free DNA, which is released because the sample was shipped, frozen, and thawed again twice. And then there's something in which has only a backbone and a very minor absorbance around here. This may be your empty capsid, and you have some species in between. And at the very end, you have a backbone, but a huge absorbance here, 260. So these may be your full capsids, so this stuff here. You, of course, do not need to rely on the visual inspection of the data and believe what I have told you just before, but you can take a intrinsically, let's say, fitted DNA and empty capsid spectrum, which means you, as said before, you dilute a sample of your empty capsids and your DNA, if you have it at hand, and collect a short span of concentrations, fit it globally so that you get rid of the non-linearity. And then you can deconvolute this. This can be all done in Ultrascan. You can deconvolute this two-dimensional data set, or three-dimensional data set actually, into the signal of capsid and the signal of DNA. And when you look here, you see here in red just the DNA signal. So everything you see here is red. It's protein. It's DNA, and in green, it's the empty AAV signal, which means capsid signal. When you then integrate your empty capsid signal, you get the ratio of your capsids, which is not depending on a single wavelength, but on the multi-wavelength data set I've shown you before. And as you could discern from the visual comparison, you can now really say, okay, this 17S peak here is my free DNA. This is empty capsid only. Then you have partially filled capsids, which contain DNA, but not as much, almost as a one-to-one stoichiometry, like the full capsids here. This is the respective Van Hall-Weisheit plot of that. And you can convert that now also into percent titer using the extinction coefficients I've described you before. And when you do that, we came up with 26% empty, 43% partially filled, and 30.6% full particles. And this we used as a benchmark for comparing the data I've shown you before for the single wavelength, but also for the experiments I'm going to show you in a second. Okay, just to recap what I've said before, a multi-wavelength experiment is really suited. It's really much like a multi-attribute method. You can not only get a single ratio of, let's say, empty and full particles, but you can also resolve very nicely, like with any velocity experiment, the partially filled particles. It reduces the uncertainty of a single or dual wavelength measurement. You incorporate data from a orthogonal second technique, UV-Vis spectroscopy, by measuring spectra. And it delivers you, as said before, capsid, ratio titer, the total OD, the free DNA. You can also look for aggregates, degradation product. So it's really useful. But of course, data analysis may take some time, and it consumes, and this is the only really hard constraint, I would say, it consumes quite a lot of sample. So in our case, we needed 200 to 300 microliters of the undiluted product. And as you may have heard, Zolgensma is quite expensive. It's, I think, 2.1 million per dose. So the question is, can we save material for routine analytics? And the answer is yes. There is a technique available for decades. You may have heard of the famous measles and Stahl experiment in the late 50s, 1958, I think, for the replication of DNA. And what you are doing is you are performing an equilibrium experiment on the salt, in this case cesium chloride, which forms a gradient. And in parallel, you have a velocity experiment ongoing because material is sedimenting from the bottom and sedimenting from the meniscus and floating from the bottom until it reaches a position in the gradient where the density is equal of the density of the hydrated material. And a very, long-standing application of that for AAVs is the purification process. So basically what you're doing with such an CSCL SE AUC experiment, you are replicating the purification process, but using it for analytical purposes. So this is here in the middle. You can see a commercial preparation, which means you can separate empty AAVs, regular AAVs, and also you have, and that's point, I think, still under discussion, what we call heavy AAVs. They may be overloaded AAVs, but that's not really sure. It's still an open question. You see that also here in the data on our actual sample. You see very nicely a homogeneous and symmetrical peak for different salt concentrations. We probed 1.34, 3.6, and 3.8 gram per milliliter cesium chloride. The empty peak is nicely separated, and you can also see it's fairly symmetrical. But when you look at these peaks here, you may assume that's partially filled, and we actually did that, and full. And there are several shoulders on the right-hand side, and I do not really know what it is. And if you are sure what it is, please tell me. I think that's still a point of active discussion. In the 80s, Already Myers and Carter discussed about that. These may be overfilled particles, but as said before, it's not definitive yet. Okay, so the good thing about that method, if we talk about methods today, time is not an issue in an equilibrium experiment. You can collect as many scans as you like. So for example, you do not go for only every second wavelength, but you can really go for as much wavelength as you want. And in our case, we collected, I think, 61 wavelengths from 230 to 290, but not in 2 nanometer steps, but really every nanometer. And what you get is a 3-dimensional data set looking like this. You may see here already from looking at the top, all of the 3 major species have a backbone, which is not surprising, so it's protein in there, but the absorbance here at around 260, 70, and 80 varies quite. So this is again your empty particles. This may be partially filled because the absorbance at 260 is a little higher than than for the full particle, when the OD here at 230 is roughly equivalent. But again, you need not to rely on what you can discern by eye. You can take out the individual absorbance spectrum just by slicing through that data set for each of the species and deconvolute that. In this case, it's shown here for the gray trace, the full particles, and it contains, as expected, roughly by molecular weight, 70% protein and 30% DNA. Okay, so cesium chloride, SE-AUC, is really handy because it involves a sample concentration step, which means you can start at very low concentrations. If you do the math, it's in the range of perhaps 5% of the material what you need for a multi-wavelength sedimentation velocity experiment. And as it's a concentration step involved, you may also look at in-process samples, for example, where the titer is often in the range of 10 to the power of 9 or 10, so 2 orders of magnitude lower than for the final product. You have plenty of time. You can scan as many wavelengths as said before. The data analysis is, compared to a velocity experiment, relatively easy because it's integration. You need, of course, to take into account, if you use a standard velocity centerpiece, the radial dilution or the increase in volume, because it's not a rectangular volume you have, so the volume changes with the radius. You need to take that into account. And what in our experiment also showed up is that you have a narrow density window, which means if you want to separate empty particles from partially filled and full ones, you have to choose a cesium chloride concentration at around 1.34, 1.36 or so. And at that concentration, free DNA still sediments to the bottom of the cell. So you cannot measure free DNA, for example, in the same experiment where you're looking at the empty-full ratio, for example. Okay, then we tested also a third kind of experiment, which is called analytical band centrifugation or analytical band sedimentation velocity, sometimes termed, where you have a special centerpiece with a reservoir on the top, where the sample and a reference buffer is filled in there, 10 to 15 microliters. You have your buffer filled into the sectors, like in a velocity experiment. The buffer has to have a higher density than the sample, otherwise it will just sediment, or the drop will, so to say, fall through the bottom of the cell. And if you do it properly, you get a band which is overlaid on the top of the sample, and then your molecules start to sediment into that buffer, which you have provided here in the chamber. And what you get is a band which moves to the bottom of the cell and rapidly decreases in OD because it spreads and diffuses into an empty buffer space. And you can analyze that very much like a velocity experiment. In our case, what we found out at the speeds we employed is you get a very similar size distribution compared to a standard velocity and multi-wavelength run. But the smaller species at 14,000 RPM they were not really resolved. So the resolution for the smaller species isn't really that good, maybe due to really rapid diffusion. In this case, you only need, as I said before, 10 microliter of the sample. It should be roughly in the range also of the final product, but it reduces the sample consumption also by 95%. So the sample consumption is roughly the same like for a cesium chloride experiment, but here you need a concentrated sample. For the SE experiment, you can also start with a more diluted sample. Of course, you can, for SE experiment, also take only 10 microliters of your concentrated stock and prepare that sample. You have both options for the SE experiment. So to sum that up, when you choose the right wavelength, the AUC techniques I've shown you before deliver you the same if you just look at one wavelength. So for example, at 230, comparing the velocities experiment, a data set from the multi-wavelength experiment, so the 230 trace, then this is here the 230 scan of the cesium chloride experiment and the 230 scan of the band forming experiment. And this is for comparison the multi-wavelength experiment, the same here on the bottom. This is the capsid ratio, and this is based on the titer if you convert the OD to titer using extinction coefficients. So you have quite some choices to analyze your AAVs with AUC. You have a standard velocity experiment. This is really good because it's recognized by authorities, but it depends on single wavelength, and it consumes quite some sample. The sample consumption is the same like for velocity and multi-wavelength AUC, but here you can measure more samples. For multi-wavelength experiment, you should usually restrict yourself to one sample because the scanning time is quite long, because you need to scan the sample for all wavelength, then comes the second scan for all wavelength, the third for all wavelength, and so on. So this takes 20 seconds approximately for the optima for one cell. This is an excellent characterization assay. We haven't submitted it so far to any authority, so let's see what they say to it. And if you look for alternatives, for example, for in-process sample or for precious samples, you have two other choices available. One is the cesium chloride experiment I've shown you before, where you have sample concentration. It takes a little longer, and no free DNA in this case is detectable, but it reduces really the sample concentration you need. So it may be good for in-process samples where you have low titer. And then you have your ABC, your band-forming experiment, where you only consume a tiny amount of your concentrated sample, but it lacks the resolution of smaller species. So free DNA, for example, or if you have degradation products, empty monomeric capsids, for example, this may be not that well resolved. So these are the caveats for it. And when we now look at lipid nanoparticles, we have briefly talked about that. Besides size and shape and then multi-wavelength, you can add a fourth dimension to your AUC experiment by changing the density of the solute. And these are the only equations I'm going to show you today. You all know the Svedberg equation. It contains this part here, 1 minus rho v bar. So the density of the solvent and the inverse density, or strictly spoken, the density increment of the analyte. And when you modify the solvent density, you can gain information about the partial specific volume of your sample. And as your sample is composed, as said before, in the case of cell and gene products of either DNA and protein or nucleic acids plus lipids, if you talk about lipid nanoparticles, you can say something about the relative composition, because lipid nanoparticles consist, as said before, of lipids which have a density close to that of water, and you have a nucleic acid in there which has a quite high density, almost twice that of water. And just to make you familiar with the concept of F over F0. It's an expression where you fit instead of, or actually the software still fits the diffusion coefficient, but it's connected to via the Stokes-Einstein-Sutherland relationship to the F over F0. These are the only things you need to remember when we go to the data. Okay, so just to introduce also these kind of samples, lipid nanoparticles. In our case, they were loaded with an mRNA. You can also use siRNA, for example. You have a aqueous phase and a ethanol phase. You mix it with a T mixer, add buffer, and end up with a particle which consists, in our case, of 4 types of lipid: a cationic lipid, which is responsible for the encapsulation of the nucleic acid, a cholesterol, which fills, so to say, the capsid. You have your neutral lipid, which stabilizes and improves the processing, upon cellular uptake. And then you have your shielding lipids, your PEG shielding lipids, which are here on the outside. And these modulate the size of the particle and also control the PK profile. And they suppress aggregation, so they are quite important. In our case, these were the 4 components involved, and the RNA had a length of 1,156 nucleotides, and the lipid composition is 45 to 44 to 92%, which means it's vastly composed of the cationic lipid and cholesterol. And the NP ratio, so the ratio of cationic lipid nitrogens and mRNA phosphates, because this is also important for the expected size, and the loading efficiency was determined to be 4.5 by CyberGold assay. And this is a cryo-EM image. But I'm focusing today on AUC data because these are the data I've gathered in my lab. Okay, so what you can do is you can alter the density, of course, by a variety of agents. The use of D2O or H218O is preferred because if you add sugars, for example, you alter the hydration shell or the composition of the hydration shell in respect to the composition of the solvent. In other words, the hydration shell may serve as just like a life jacket. The protein or the particle is floating. So this is called preferential solvation or preferential interaction in the case of denaturing agents. So you are changing also the apparent V bar if you use, like, for example, sugar or so to measure the V bar. That's the reason why water isotopes are the preferred mode of doing that. And when we did that in our very first experiment, we had the empty particles at our hand, the empty lipid nanoparticles, and the filled one. And the filled one you can see here in green in PBS. It has a positive sedimentation coefficient, so it sediments in water, or PBS. And the empty particles already starting here in red float already in PBS, and with increasing amounts of D2O, we went up to 25%. You can see here 5, 10, 15, 20, 25% of D2O. It really floats. And with that, we went on using a density matching module implemented in Ultrascan. And what you can do there is you load your distributions gathered at different densities. And what you can compute from that is the density or V bar distribution of your particle. And what you can see for the empty particles is it has a part here in the middle, or the majority of the material has a density which is centered around 1.014 centimeter cube per gram, which means the density is lighter than that of water. And it has a tail on the, let's say, floating and the sedimenting end. So there is a little bit of material sedimenting in there, and some stuff which is has even higher negative sedimentation coefficients, but we went on for the simplicity of the calculation just with the middle part and took this value for the density as the density of our empty particles. And then we repeated the same experiment with the full particles at 20K RPM and probed from 0 to 10% in 2% steps. This is actually one of the later experiments, I have to admit. And the first experiments, we went up to 90% or so. And yeah, the particles just floated. So after the first scan, they were completely gone. So we worked with these D2O concentrations, and the dip here in the middle is just at 0 because flotation is not defined. And we did the same like before, and what you can see is the distribution is not centered about one value with a tail on both ends, but it's a more continuous distribution for the full particles in contrast to the empty particles. And when processing the data, we had, this was our primary goal of interest, a V-bar distribution, so partial specific distribution of our full particles, because this distribution depends heavily on the, or depends on the composition or the relative amount of RNA and lipids. And what we did is we used the V-bar of the empty particles I've shown you before, and the tabulated value for RNA and PBS, and calculated also using the molecular weight distribution you get out of that, the molecular weight as a relative composition of lipid and mRNA at each point of the distribution, divided the molecular weight distribution by that, and multiplied divided the molecular weight at each point with that of the mRNA. Just to give you a graphical overview, because I've explained that sometimes and people looked at me and said, okay, still didn't get it. So as said before, this is the V-bar distribution. We take the value of lipid and the V-bar of RNA. You get a relative fraction of percent lipid and percent RNA. And as you can already see from the density of the full particles, they are very close to water, which means they consist almost exclusively of lipids. And when you look at the numbers, this is actually true. The distribution is quite narrow. It ranges from 92% to 96% of lipids. So they vastly contain only lipids. And when you now add the molecular weight distribution and calculate the relative molecular weight fraction, so to say, of this distribution, and use the known molecular weight of the RNA, you end up with a distribution like this. You see here a stepwise increase in the orange trace here. This is the S value. It increases, not linear. We fitted it with 512 grid points, so quite fine. But you see here a stepwise increase. And if you calculate the number of mRNAs, you see here it goes from approximately 1 to 2 to 3 to up to 10, and has an average of 4.6 mRNA copy numbers per lipid nanoparticle. So this is what we came up with, and what we also reported back to the collaboration partner. And what we did finally, we also had a look into the main peak I've shown you before. We measured it at with 3 independent experiments and fitted it with, as said before, 512 grid points, and looked for the S values. When you look here at this graph, you see in all 3 experiments, you have an increase of approximately 5S for each species, which is, yeah, approximately that what you also expect as an S value for the free mRNA. Of course, that's only a very, it's another hint, it's not a proof, but we were quite confident that indeed the mRNA lipid, the mRNA copy number is in the single digit range and not, let's say, 50 or so. So to sum it up and open for questions in a few minutes, With the supercomputer-aided analysis of AUC data, you can really fully exploit the experimental capabilities you nowadays have. You have the first dimension, which is the sedimentation coefficient distribution, which is great for aggregation quantification. You have a second dimension where you can fit in SETFIT or Ultrascan, for example, the molecular weight of individual species. Then you have a third dimension where you can collect dozens of wavelengths and have a multi-wavelength experiment globally fitted instead of a single pair of wavelengths or just one wavelength. And if you play around with the density, you also can add a fourth dimension, which allows you to exploit the density difference between different materials which your cell and gene product may be composed of. So AUC proves to be really a powerful tool for the third generation of pharmaceutical products after small molecules and proteins, so to say, genetically engineered proteins or recombinant proteins, as you may call it. And it has led, I think, to an increase of AUC users and instruments, and which is really great now, and perhaps somebody's also in the audience, there's also GXP compliant control and data evaluation software, especially evaluation software available, which is Ultrascan and BASIS. So AUC may be even more relevant in the future as a QC method for AVs, for example. And with that, if you are interested in what I've told you today in more detail and also want to have a look at the numbers and the calculations, you can look into these two papers. And from now, I'm happy to take any questions.
Thank you, Dr. Bepperling, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for today. Let's go ahead and get started. Our first question asks, could you also quantify the number of siRNA molecules in a siRNA LNP?
I think so. There are some limitations. The first one is siRNAs are often conjugated with carbohydrates, so the density may not be that of just the nucleic acids, and also the extinction coefficient cannot be simply calculated from sequence because it may not really average out like it is for a longer mRNA. But in principle, yes, I would expect you may have a larger error. Also because the number of molecules is usually higher. So as seen here, we are talking about single-digit copy numbers for mRNA, but for siRNAs, as far as I remember it from literature, range of several hundred to even 1,000 molecules per vesicle.
Great, thank you. Our next question asks, is Ultrascan available to be purchased?
I think so, yeah. We have purchased a system called Ultrascan in a Box, where you have your own in-house server, and it allows you to store your data not in the cloud, so to say, but locally. And I think if you're interested in pricing, you may contact Boris Demler directly, but it's commercially available.
Wonderful, thank you. Our next question that we have here says, how much better can you characterize for partially filled AAVs by MWL AUC compared to measuring at 2 wavelength, 260 and 280 nanometers?
So hydrodynamically, I would say not much better, even a single wavelength, because you have more scans. Maybe if you look at the, let's say, species separation, more preferable. But if you think about want to know how much nucleic acid in there. Multi-wavelength is, of course, much more preferred. Yeah.
Wonderful. Thank you. All right, our next question here says, the LNP characterization is exciting. Thank you for showing the data. How important is AUC in your organization for LP characterization, and which other methods do you use for the characterization of LNPs?
What I can still say, because I'm not part of Novartis anymore, is cell and gene department has, I would say, 90% of the centrifuges which Novartis owns. So it's really an important tool for that. But yeah, I'm not Novartis anymore. I wouldn't talk too much about what other methods they are using. At Sandoz, we are working on more. If you talk about lipid particles, we are more working on generics like liposomes filled with, for example, cytotoxins. So it's more small molecule business.
Thank you. All right, our next question here says, how do you organize the supercomputer-aided analysis, and can you explain how that supercomputer compares to a PC?
This depends, of course, what you purchase. So what we have is a two systems, actually, one EPIC and one Ryzen with 128 and 61, sorry, and one with 64 units. If you compare how long it takes in terms of time, I would say if I have a standard sedimentation velocity experiment on my desktop computer with ultrascan, which can be done, it takes perhaps 30 seconds to 1 minute or so, and on our supercomputer system, I would say 5 seconds. So roughly, I would say at least 20 to 30 fold. It's absolutely.
Oh, that's great. Thank you. All right, our next question here says, how do you handle the dynamic range of absorbances at different wavelengths when collecting multi-wavelength data? It is preferable to keep A230 in the ideal range while having lower ranges for 260 or 280.
Right, that's an excellent question. And this is also something where we put quite some effort in there. In the first experiments, we went for a high 260 absorbance because that's what we did before for single wavelength. But yes, then of course at 230 you have an OD of 1.8 or so, which is not what you like. And the data I've shown here and also the data we have published are such as described by the colleague. We went for an OD of 0.8 around for A230, and consequently, of course, I only have an OD of 0.3.
Great, thank you. We have a lot of great questions coming in. Again, if you have questions for Dr. Bepperling, please submit them in the Ask a Question box located in the lower left corner of your screen. We'll go ahead and move on to our next question here. That question asks, what may be the advantages of AUC for the analysis of short oligos like ASO, siRNA? Do you have any experience in this?
So if you're talking about free oligos, so just formulated in water often, or just least the siRNAs and antisense I've been working on were just formulated in water. The big advantage is, compared if you think about peptides and other small molecules, that the V bar is quite high, so you can really nicely separate them. When working on peptides, I always have the problem, the monomer and the dimer and so on, they are not separated. But for oligos, even if you only have, let's say, 20 residues, this is really nicely separated. It has an S value, the monomer, of about 2.5 or so. So that's quite good, yeah. So AUC is quite for that.
Wonderful, thank you. All right, our next question here says, in SE experiment for AAV, can the tail and the full species be related to some interactions between the AAV and cesium chloride? Cesium, sorry.
That's a really tough question because, as I said before, I'm not really sure what high density species are. I did quite some, also in front of the webinar, some literature research and also for the publication. And what the evidence so far suggests is that the full particles still have the same of A260 to A280. So they aren't much different in terms of lipid as of nucleic acid. Yes, these species only show up in the cesium chloride gradient. You only separate them in a velocity experiment. So yes, indeed, cesium chloride may induce some of that change or, yeah, promote, let's say, the occurrence of these. But I don't really know for sure right now.
Wonderful. All right, our next question here asks, is Ultrascan going to replace SEDFIT and Gussie for the analysis phase?
Oh, a deadly question. No, I wouldn't go that far. And at least in my lab, I also encourage, if I have new colleagues, to really get familiar with all the software packages which are available. I know there is content and dispute going, but I do not really take a stand on that. For data management and also having an overview, I really so controlling your instrument, it's a base, but nevertheless, SEDFIT and AUC, and also we haven't mentioned today Walter Stafford Sedanal, they have their individual strength for certain systems. For example, Sedanal has a really cool model editor. So, no, I wouldn't say any software will replace or really fully replace the others. So they all have their value.
Great, thank you. And it looks like we have time for one more question here today for you. That question asks, how many samples have you analyzed during one multi-wavelength experiment?
For velocity, only one. For the length experiment, 3 or 7, depending on the rotor.
Excellent. Thank you, Dr. Bepperling. I wanted to ask, do you have any final comments for our audience today?
No, but I was very delighted to see that we had an intense, also I think 13 questions in total, which is really nice and shows that AUC has really a bright future for cell NLP analytics, I would say.
Absolutely. Thank you again, Dr. Bepperling, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. I want to remind everyone that there will be a survey that pops up at the end of this webinar. If you could please take a moment to submit your responses before you leave. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.